So, like I was saying, that the ovules are of the following types. The first one is the orthotropous, where the micropyle, the funicle, which is the attachment of the ovule with the ovary, that is in a single line. So the chalaza is this part, the micropyle and the funicle, they lie in a single straight line. That is orthotropous. And when the funicle is forming a circle, that it is wrapping around the ovule and the micropyle is still pointing upwards, that is known as cercinotropous. And when it is completely inverted, like the micropyle comes near the funicle, then we call it anatropous, which is this one. And here, when the micropyle and chalaza are forming a right angle with the funicle, then we call it hemianatropous. And this one is when it is slightly curved and the embryo sac becomes like a horseshoe shape, then we call it amphitropous. And the last one, where there is no such angle and it is extremely curved and bent, then that is known as a campylotropous. So these are the types of ovules. Next is the development of female gametophyte. So in this case, the first thing is what is megasporogenesis. So in case of male, we have already seen that it is microsporogenesis, and now in female we are calling it megasporogenesis. So obviously, because of the size of the spore, the female spores are much larger, so we call it megaspore, and the male ones are smaller, so we call it microspores. So in this case, the female gametophyte starts developing from the megaspore mother cell. So as you can see that there is within the nucellus. So this is the nucellus portion, and the ovule, the majority of the tissue in the ovule is nucellus only, and the rest of it is integuments, which are just the protective coverings. So inside the nucellus there is a megaspore mother cell. So this is the large cell, as you can see, and here the megaspore mother cell is undergoing meiosis. So in the first meiosis, as you can see that there are a number of haploid megaspores. So with one complete meiotic division, there will be 4 haploid megaspores. Out of those 4, only 1 megaspore will be functional and the 3 will degenerate. So the 3 megaspores will degenerate, and therefore the 4 megaspores together are known as a megaspore tetrad. Similar to what happened in the microspore tetrad was formed, and here also megaspore tetrad. But the only difference is 4 microspores were viable and they were alive. But in case of megaspore, only 1 megaspore is surviving and functional, and the other 3 are degenerating. Okay, so definitely megasporogenesis is the process of formation of haploid megaspores. This process is known as megasporogenesis because it is formed by the meiotic division of the diploid megaspore mother cells inside the megasporangia, in this case, which is the ovule. So next, the megaspore, the haploid megaspore, undergoes repeated mitotic divisions and gives rise to a 8 nuclei stage, or the 7 celled but 8 nucleated embryo sac. So the embryo sac is known as the megagametophyte or the female gametophyte. Now in this case, the first mitotic division will give rise to 2 nuclei from one functional megaspore, that is this one. And in case of second mitotic division, 4 nuclei will be formed, and the third mitotic division will give rise to 8 nuclei. So the 8 nucleated stage is achieved. Next, cells are formed around the nucleus, and it will give rise to 2 synergids over here and 1 egg cell, which is here. 2 polar nuclei will come in the central part of the embryo sac, and at the other end there will be 3 antipodal cells. So antipodal cells are basically here for providing the nutrition to the embryo sac, and the synergids are basically to direct the pollen tube so that the pollen tube can enter through here. This is the micropylar end, and the micropylar end, like I already said, there is a gap in the integuments, so the pollen tube can easily enter from here. It can destroy one synergid and it can come in contact with the egg cell. So this is the basic 7-celled, 8-nucleated embryo sac, because these two polar nuclei, they come together and form a single cell. So the very important term here is the double fertilization. So what happens when the sperm or the pollen tube enters the ovule? So first the pollen tube is adhering to the stigma, which I already mentioned, and it contains two cells, a generative cell and the tube cell, or the vegetative one, which is now the tube cell. And the sperm, the two male gametes, are now traveling across the pollen tube and reaching the ovules. So inside the ovule, it enters through the micropyle, and through the micropyle, when the pollen tube is entering, it is releasing the male gametes inside the embryo sac. So here one male gamete will fuse with the egg and it will form the diploid zygote, and the other sperm will bind with the two polar nuclei, and it will give rise to a triploid endosperm. A 3N cell will be formed, and this will be the precursor of the endosperm. This will become a future storage of food for the embryo. So this process is known as double fertilization. One is syngamy, or the generative fertilization, that is happening by the fusion of male gamete and egg, giving rise to a zygote 2N, and that will ultimately give rise to an embryo. And the other one is the triple fusion, or the vegetative fertilization, which is by the fusion of one male gamete and two polar nuclei. The two polar nuclei are together called the secondary nucleus, and these three together gives rise to primary endosperm nucleus, or 3N, which is triploid, and will give rise to endosperm, which is the nutrition providing tissue of the embryo. And ultimately the ovule will become a seed and the ovary will become a fruit. So these two events happening together is known as double fertilization, which happens in case of angiosperms only. Next, if we look into the life cycle of angiosperms, we will find that there are two important phases: the sporophyte phase, which is the diploid phase, and the gametophyte phase, which is the haploid one. So if we start with the mature flower, then so this flower obviously has the male reproductive unit as well as the female reproductive unit. So the male reproductive unit is like this: the microspore mother cell, from here the microspore tetrad, the 4 microspores getting released, the generative cell and vegetative cell, or the tube cell, getting formed, then 2 gametes from the generative cell, and then the pollen is developing a pollen tube when it reaches the stigma of the female reproductive unit. Inside the female reproductive unit, there is the megaspore mother cell, which divides in meiotically and give rise to 1 functional megaspore, 3 degenerating megaspores, and this megaspore is ultimately forming 8 nucleated embryo sac, and inside this embryo sac there are 3 different kinds of cells: the egg cell, the synergids, the antipodals, and 2 polar nuclei. So all these are present here, and then the pollen tube is coming from here and reaching through the micropyle. Ultimately this will happen, double fertilization will happen, triploid endosperm nucleus will be there, the endosperm is getting dividing, multiplying itself, and the seed is formed. This is the seed which contains the embryo, the endosperm together, and these are seed coats. Then the embryo will develop from here, and again it will give rise to a sporophyte. So this is the sporophytic generation. The 2N part is the sporophyte, and from here it is again gametophyte, that is the N. So the gametophyte arises when the cells of the sporophyte undergo meiosis and produce haploid cells, and the sporophyte is the typical diploid plant body that we are usually seeing like this. So next is the establishment of symmetry in plants. In case of plants, there are 4 different whorls. Like first whorl is the calyx, which are made up of sepals. The next one is the petals, and it is known as corolla. The third one is the male reproductive unit made up of stamens, which consists of anther and filament. And the fourth one is the female reproductive unit, which is known as carpel, and it consists of stigma, style, and ovary, and ovules within the ovary. And all these whorls are arranged in a base or thalamus called as receptacle. So the arrangement of the leaves or flowers around a plant stem or even in the whorls, this is known as phyllotaxis. And in case of spiral phyllotaxis, like you can see that the elements grow one by one at a constant angle from the other one. So one is at a constant angle from the previous one, and this kind of spiral phyllotaxis is usually done by a definite divergence angle, which is known as golden angle. It is usually 137.5 degrees. So at this angle, usually the elements are arranged one after the other. Next is the meristematic tissue. So obviously in the cells, in the embryo, in case of plants, embryogenesis, meristematic tissue is very important. Meristematic cells are basically the cells which are at the tips, growing tips of the plants, like the shoot tip as well as the root tip. And these are undifferentiated or slightly differentiated cells, and they are considered the stem cells of plants because they can give rise to multiple number of organs of the plants or multiple types of tissues. And they can form various parts of the plants. These are lateral meristems are located around the stem, and they can increase in the thickness or the diameter. And intercalary meristem are present in the nodes where from the leaves or the branches can develop. And of course the apical meristems are present in the shoot and root apex, and from here the length can increase. So the cell divisions in plants are of the following types. Like in case of there are anticlinal divisions, periclinal divisions, and transverse divisions. So transverse is when the division of plane is like this. There is a horizontal plane of division. And in case of periclinal and anticlinal, both are vertical plane of division. But the difference is that in case of anticlinal division, there is increase in the thickness, and it is perpendicular. The growth is perpendicular to the division of plane of division. And in case of periclinal, the increase in length and the growth is parallel to the plane of division. So that is the difference. As you can see that this plane of division is like this. Cells are dividing in, and there are two cells from one cell. And here there are two cells from this cell by this kind of a division. So this is periclinal and this is anticlinal. Here by means of a cube, that same thing is determined. So seed formation in plants or embryogenesis, like I said, that the seed contains two major tissues, the endosperm and the embryo. So endosperm is the nutrition providing tissue, and the embryo contains the cotyledons, the seed cotyledons, and the plumule, the radicle. Plumule is the shoot tip and the radicle is the root tip, and it is covered by the seed coat or integuments. So this embryo development has a lot of cell division, cell differentiations, and this embryo development is bipolar. That is, it will grow in two poles. Like one pole where the shoot is there, it will be negatively geotropic, that is, it will grow against the gravity, and there is a radicle which is the root tip, which will grow positively geotropic, that is towards the gravity. So these are at opposite ends. So therefore embryo development has to be bipolar, that is, the development is directed towards two different poles. And this is, if we consider this as the zygote that is formed after double fertilization in case of angiosperms, this zygote after first division, it will give rise to a smaller apical cell and a larger basal cell. So from the apical cell, the major part of the embryo will be formed, and the basal cell will give rise to the suspensor cells, which are responsible for connecting this zygote with or embryo with the endosperm for providing the nutrition. So this part is known as a globular embryo, and from here, like you can see that a heart-shaped embryo is formed. So this heart-shaped embryo contains cotyledons. There are two cotyledons, which means it's a dicot, and this is the shoot apical meristem, like I already said, that from where the shoot tip will be formed. And this is the root apical meristem, where the root tip will be formed. And this, after after multiple divisions, there is a change of shape, and it is known as a torpedo embryo stage. So from heart shape to torpedo shaped, and then after many such steps, it gives rise to a mature seed, where there is the root apical meristem, there is the shoot apical meristem, and there are two cotyledons, and are ultimately covered by a seed coat. Okay, so next is seed germination. So in case of seed germination, germination, like we already know, that that is the growing of a new plant from a seed. That is known as germination. So it is basically a switch from an anabolic phase of nutrient accumulation. Like I already said, that the seed has to store nutrition for the embryo inside, and then that is known as the anabolic phase. And then it has to switch to a catabolic phase, like when the embryo needs to consume that nutrients and become active and should germinate. So this is known as germination. And in between these anabolic and catabolic phase, there is a quiescent period or a dormant period, and this dormancy is usually broken by certain factors. Like factors which affect germination can be external or internal. In case of external, these are abiotic factors like light, temperature, water, oxygen. We all know that these are very important for proper seed growth, germination. And in case of internal factors or the biotic factors, there must be some nutritionary factors which are inside the seed, which needs to be in proper amount for the proper development of the embryo, and only then the seed will be viable, and then it can give rise to a new plant. As well as the growth regulators or the plant hormones, which are auxins, gibberellins, cytokinins, abscisic acid, etc. So these are very much important for regulating the growth of the embryo. If these are not present in optimum concentration, then the germination will not happen. So these are the major factors determining germination. So in case of monocot, like you can see that there is only one cotyledon here inside the seed, and the endosperm is here, and there is the hypocotyle and there is the epicotyle. The hypocotyle is then surrounded by radicle, and there is the seed coat which is covering the entire seed, and it is fused to the ovary wall. So from here it is, in the case of monocot germination, the epicotyle will start emerging, and there will be the hypocotyle, which is the connection of the radicle and the shoot apex. And from epicotyle, the shoot is usually formed, then roots develop, and the first leaf starts forming. In case of dicot also, there will be two cotyledons and a radicle and a hypocotyle connecting the radicle with the epicotyle, and ultimately the seed coat covering it all. And this seed coat will burst, and the hypocotyle will start developing, and the root will start form, and ultimately the first leaves are likely. So what are the steps where how the seed germination occur? So first there is the imbibition, that is the swelling of the seed, the accumulation of water, absorption of water, and that will ultimately make the seed coat burst, and so that the embryo can develop and it can come out of the seed. Next is respiration. Oxygen has to reach there, and the effect of light, we all know that without light this cannot happen. And there will be mobilization of reserves and the role of growth regulators, I already mentioned this, and development of embryo axis into seedling. So the axis, like the shoot and root apex, that needs to be determined properly so that the seedling can start developing. So this is all for today's video, and it ends the gametogenesis and fertilization part. And next we will start with section C. Thank you.